So in this video we're going to think about where proteins end up in the cell and how the cell controls getting the right proteins in the right place. So obviously from the nuclear genome we need to make proteins that go to all sorts of different locations within the cell. And that needs to be really tightly controlled. You don't want to end up with proteins that are supposed to be in the mitochondria ending up in the Golgi, for example. So we need to have mechanisms in order to control this. And then the way that the cell does this is what it does is it effectively gives its proteins little postcode sequence to tell it where it should be. So if we think of a protein here. Okay, so there's my polypeptide. We've got the N terminus and the C terminus. So this is my protein. That protein will contain several bits of information in it. So let's say that the middle bit of the protein sequence is the bit that actually encodes the enzyme activity. Okay, so we've got not all. So if it's an enzyme, maybe only about half the protein is actually encoding that enzyme. You might have regulatory sites. So you might have a bit of your protein there that is, let's say, has got calcium binding site in it, so that might make it a calcium dependent protein. But what you also have in most proteins, so any protein that isn't going to be in the cytosol, you will have some sort of target sequence. So it could be right at the beginning of the protein, it could be in the middle of the protein, it could be the end of the protein. But that target sequence tells the cell where the protein should end up. So effectively, it does work like a postcode. So you can imagine there's a particular sequence in there, a particular sequence of amino acids that's effectively, it's a marker that says, please take me to the nucleus, or please take me to the mitochondria. And there are specific sequences that do that. So for example, if at the C terminus, at the end of the protein, you have the sequence K-del, that's using the single amino acid codes. If you have the sequence K-del right at the end of your protein, that tells you that it should end up in the endoplasmic reticulum. Okay, so some sequences are quite short, some sequences are long. So K-del's quite a short one. We've got those 4 amino acids, but if we see them at the C terminus, it doesn't work at the N terminus, it only works at the C terminus, then that tells us that that protein ought to end up in the endoplasmic reticulum. And we can do experiments to test this. So let's imagine this experiment. So let's say that we've got a protein, just draw out my protein like that, so simplify its structure. So let's say we've got a protein that at the beginning of that protein, we have a signal sequence that tells it to go to the mitochondria. Okay, so how could we test how that sequence works? Well, what we could do is to first of all attach that protein to GFP, which is green fluorescent protein. So if we look at that protein under a fluorescence microscope, then what we should see is quite a sort of dotty pattern. So we should see GFP in the mitochondria. Okay, so that would be our control in our experiment. But what we could then do is to deliberately mutate the protein. Okay, so we keep it hooked up to GFP, but this time we have removed the signal sequence. So the rest of the protein is unchanged. We've still got green fluorescent protein there, but we've now removed that mitochondrial targeting sequence. So what we should then find, if we look under the confocal, is that GFP would be in the cytosol. So the cytosol is kind of the default location. So if there are no signaling sequences in our protein, then it should end up in the cytosol. So if we remove that signal sequence, then we should see a change in the pattern of GFP. What we could also do is an experiment to add that sequence to the beginning of another protein. So let's say it's a protein that should be in the cytosol. We could add that sequence to another protein and see if that changes its location. So that's quite an important type of experiments to check how a protein is trafficked within the cell. So we know quite a lot about trafficking routes, and we know that there are 3 routes in the cell for nuclear encoded proteins. So obviously the mitochondria and the chloroplast have genes of their own. So genes that are expressed in the mitochondria or the chloroplast, they will stay in the mitochondria or the chloroplast. What we're talking about is the genes that are in the nucleus, which are translated in the cytosol. So if you remember in gene expression, the mRNA gets chucked out of the nucleus and goes into the cytosol. So by default, our nuclear encoded proteins are going to be made in the cytosol, but there are 3 main locations that they might need to go to, each of which would need its own postcode or targeting sequence. Okay, so where are these 3 locations? Well, the first location is actually into the nucleus. Okay, so we need proteins in the nucleus, we need histones, we need transcription factors, we need all the DNA binding proteins. And although those are encoded in the nuclear genome, they're translated in the cytosol. There are no ribosomes in the nucleus, the ribosomes are in the cytosol. So if we need a nuclear protein, we actually have a way of that protein getting back in to the nucleus. Okay, so how does this system work? Well, you probably know so we know that the nucleus is a double membraned organelle, but it does have gaps in its membranes, and those gaps in its membranes are what we call a nuclear pore complex. So there's quite a lot of different proteins there that they make a sort of basket structure at the membrane, and these are sort of tendrils coming off to kind of catch proteins in the cytosol. So we have a nuclear pore complex, and that can control which proteins go in and out of the nucleus. Okay? So if we are going to be a protein that goes into the nucleus, so we might have a protein like this. So there's a bit of protein, it's a bit in the middle. So I've got a protein like there. And what we would have in a nuclear protein is one of these signalling sequences. So that would be what's called an NLS, because a nuclear localization signal. And that can be anywhere in the protein. It can be at the N terminus, it can be at the C terminus, it doesn't matter where it is. Okay? But that nuclear localization signal, we have other proteins that are able to bind to it. And when those proteins bind, that's allowed to go through the nuclear pore complex and into the nucleus. So there's our protein, it's made it into the nucleus with its nuclear localization signal. Okay. This is a post-translational mechanism. So the ribosome has been gone, done its thing. We've just got our protein. It's got a nuclear localization signal that is bound to by other proteins, and that gets it into the nucleus. There are also proteins that have another signal. So this is an example of a protein that needs to go into the nucleus and stay into the nucleus. But you can also have proteins, so I'll just draw another one here, that have both a nuclear localization sequence and they also have a nuclear export signal. So these sorts of proteins, which is an NES, so these sorts of proteins are actually able to go in and out of the nucleus because they've both got an import signal and an export signal. So these sorts of proteins can dynamically change whether they are inside the nucleus or not. Okay, and that might be quite important for a signaling protein, for example. So you might have a protein that when it gets activated, it can move into the nucleus. When it gets deactivated, it moves out of the nucleus. So those sorts of proteins that have both an import and an export signal are often used in cellular regulation. Okay, so the nucleus, we can either have just straightforward imports, so that might be a histone protein. Okay, so histones don't need to come in and out of the nucleus, they can stay in the nucleus and package the DNA, whereas this sort of protein might be a signalling protein, or it might be a transcription factor. So some transcription factors are sometimes in the cytosol and sometimes in the nucleus, and obviously where they are depends on whether they're activating gene expression or not. So nucleus, we go through the nuclear pore complex, it's a nuclear localization signal, and sometimes we have an export signal as well. So second location is either the mitochondria or the chloroplasts. So they have kind of very similar systems for those two organelles, but they are independent of each other. Okay, so a mitochondrial targeted protein, so let's just draw it like that. Okay, a mitochondrial targeted protein always has its target sequence at the N terminus, which is the start of the protein. Okay, so for the nucleus one, it didn't matter where it was in the protein. For this one, it's always at the start. Okay, so there's a particular sequence that says, I am a mitochondrial protein at the beginning of it. Okay, so then what happens, so if we've got our mitochondrial membrane, so this is the cytosolic side, this is the mitochondrial side. We then have a complex of proteins at the membrane. We have one on the inner membrane and one on the outer membrane, and they are quite a big complex. So what they do is to feed the protein through. So the protein unfolds, and it only feeds proteins through that have that target sequence. Okay, so the protein is going into the mitochondria. Okay. And then this one's an irreversible step. So what happens when you get into the mitochondria is then there's some enzymes that cut off that mitochondrial targeting sequence. So the final protein, it's then allowed to refold. but the signal sequence has been cut off. Okay, so we've got signal cleavage. So that means that for these mitochondria and chloroplast versions, that the mature protein, so let's imagine that's a protein involved in the Krebs cycle, and that protein will be shorter than we might expect from its gene sequence. So we'll cut off a few amino acids at the beginning, so it might be sort of 30, 40 amino acids. So our protein is smaller than it was before because we've cut off this signal this signal sequence. We don't need it again. This is unidirectional. It's irreversible, which makes it different to the nuclear one. Okay, so the nucleus version of this was potentially reversible. Okay, because you can go in and out of the nucleus. The mito or the chloroplast version is irreversible because we cut off the signal peptide. If you want to get targeted to one of the locations within the mitochondria, so you want to go into the mitochondrial inner membrane, or you want to go into the thylakoids of the chloroplasts, there are in fact other signal sequences behind it. So you might have the original N-terminal sequence there, but then you might have a second sequence just behind it. And that second sequence might take you to and if it's in a chloroplast, it might take you to the thylakoid, for example. Okay, and that would also get cut off at the end of it. So, so there we've got that one, and then when that's in its final position, then we actually get to our mature protein. So we've got a second sequence. So in this case, that would now be the mature protein that was sitting inside another compartment. So the last version is to go via the endoplasmic reticulum. And the ER is the first step for targeting for lots of different proteins in lots of different locations. So proteins that are going to the Golgi, to the plasma membrane, to the vacuole, to the lysosome, lots and lots of different locations. The protein goes via the endoplasmic reticulum first, and then there's a series of vesicles that takes it to its final destination. So there's lots of proteins that go via the endoplasmic reticulum that don't end up in the endoplasmic reticulum. They end up going somewhere else. And this is different because this is a co-translational mechanism. So both the nucleus and the mitochondria version were post-translation. The protein had already been made. Whereas this mechanism is co-translational. It happens during the process of translation, so the ribosome is involved. Okay, so what happens here? So here's our ribosome. So the ribosome has got a piece of RNA and it's starting to make a bit of protein. Yeah, but that first bit of protein that it makes, it realises has got a signal sequence on it. Okay, so we have, just thinking what colour to do this in. We'll do this in this orangey colour. Okay, so we've got a signal sequence, and that's been made. And signal sequences for the endoplasmic reticulum are usually quite hydrophobic. Okay, so they've got lots of hydrophobic amino acids in them. So what happens when this signal is first made is it's hydrophobic. So it doesn't like being in water very much. So what happens is another protein called the SRP comes and sticks to it. So SRP is also quite hydrophobic, so that comes and sticks to the signal sequence. And what that SRP does is to temporarily stop translation. So basically what the system is saying is, hang on, we've got something hydrophobic coming out here. That doesn't feel like a good idea. That feels like it's probably supposed to be something in the membrane rather than something in the cytosol. Let's stop translating it and let's take it to a membrane and start translating it in a more hydrophobic environment. So if we've then got, so now that's the cytosol and this is the endoplasmic reticulum. So this whole complex goes over to the endoplasmic reticulum where there are receptors sitting on the membrane and there are translocators. Okay. So our ribosome and our SRP come and bind, so that's the SRP receptor. So we get recognition there. Once we've had recognition, then translation restarts, and that protein ends up being fed through a translocator. Okay, so this is the translocator, which is a big complex of proteins that basically feeds the protein through the membrane. And there's two things that can happen there. One is we end up with our protein inside the endoplasmic reticulum, and again we get that cleavage. So the signal gets cut off. Okay, so the protein's now inside the endoplasmic reticulum. Or what we can have is sometimes, again depending on another sequence, so let's say we've got a blue sequence in there, so let's say we've got that on our protein. So if we've got a sequence in it, it might tell the protein to stay in the membrane. Okay, so if we've got a membrane, it's what's called a stop transfer sequence. So if there's no further sequences in it, it will go into the endoplasmic reticulum. If there's a stop transfer or membrane sequence, then the protein will actually stay in the membrane. So all membrane proteins pretty much go through this endoplasmic reticulum route as they get fed through the endoplasmic reticulum membrane, but they actually get stopped in the membrane because they've got another sequence that says, please keep me in the membrane. So there's been a lot of stuff going on there, but let's just recap it. Okay, so all of this is because the proteins encode their own targeting sequences. So within the sequence of the protein, there's a message that says, I am a mitochondrial protein, please take me to the mitochondria, or I am an endoplasmic reticulum protein. We know that that works because we can do this experiment with the GFP, so we can swap these sequences about, so we can get rid of sequences or we can swap them to other proteins, and that demonstrates that the sequences work. We've then got three major routes. So our first route is via the nucleus. So we have a nuclear localization signal. This is post-translational, nothing to do with the ribosome, and this can be reversible if you've got both an import and an export signal. So things like transcription factors or signaling proteins can go between one compartment and another. Mitochondria and chloroplast mechanism is irreversible. We have signal sequences at the beginning, so we have a mitochondrial targeting sequence that then goes through a complex and that gets cleaved off. Okay, so if we've got one signal sequence, we'll end up into the matrix or to the stroma. We might have a second sequence that takes us to another location. And then the final mechanism is to go via the endoplasmic reticulum, and this is also an irreversible mechanism. Because again, we have a signal sequence that gets cut off. So we start with our signal sequence. We have the signal recognition particle comes in. We stop translating. The ribosome physically moves to the membrane. And then we restart translation and push the protein either into the endoplasmic reticulum lumen or if we've got a stop sequence, then we put it into the membrane.